Hello, everyone, and welcome to today's live broadcast. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Leica Microsystems. For more information about our sponsor, please visit their site at leicamicrosystems.com. So let's get started. I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click the send button. If you have any trouble seeing or hearing this presentation, simply click on the support tab found at the top right of your presentation window or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd now like to turn it over to our presenters, Dr. James Marr and Dr. Vikram Kohli, for today's presentation. Welcome, gentlemen. You may now begin your presentations.
Hello and welcome to today's webinar by Leica Microsystems. My name is James Marr. I'll be joined with my colleague Vikram Kohli. Together we are advanced workflow specialists for the North America region. As we go through the talk today, him and I will switch back and forth for whoever is talking. We're going to discuss our Thunder imaging platforms and how they decode 3D biology in real time. So biology today spans many different sample types. These can be monolayer cells to three-dimensional cell cultures, tissue sections, and even model organisms. These can be fixed and live. So at the top right, we have an example of cell migration. And what we're going to focus on today is our Thunder imaging platform and how it increases signal and noise, gives you better contrast for your images, and allows easier imaging of thick samples. These are a few examples highlighted here. You can see on the left is the Widefield image, and on the right is the Thunder image. And the goal of today's talk is to really explain how Thunder is applying to your sample so you get a better idea of how it improves your imaging workflow. So at Leica, we have a broad suite of products that really support you from cell culture to your advanced imaging needs. So we have systems for looking at cell culture. We have sample preparation based around our stereoscopes and upright and inverted microscopes, say our laser microdissection microscope. We have products available for routine imaging and histology based on our upright and inverted microscopes. We, of course, have our Thunder imagers that we're going to discuss today. We also have confocal platforms based on our new Stellaris that launched last year, as well as correlative Cryo EM solutions for doing electron microscopy and fluorescence imaging. Across many of these products, we have a software suite that is supported on all of them called LESX. This is so that when you switch between products, you're not switching your user interface, you're keeping it very streamlined in the same user interface. So with these products, we want to really be able to support the researcher in a very broad sample type. So with our Thunder imager, what kind of samples would actually benefit from them? So researchers have always looked at 2D cell cultures. Recently, it's been expanded to 3D cell cultures like spheroids, neurospheres, and even organoids and microtissues. Researchers also look at tissue sections, either through vibratome or microtome or other ways that can result in thin or thick tissue sections. And they also look at whole model organisms, things like Drosophila, mouse embryos, zebrafish, and C. elegans. So with all of these different sample types, Widefield has mainly been restricted to the thinner of those. The reason for this is Widefield has a lot of inherent out-of-focus light due to the way it's collected. What Thunder is meant to do is to bridge the gap and really allow researchers to image thicker samples than what regular Widefield would allow. So the way we do this is by removing the out-of-focus light or the background. And we've mentioned this a few times. So what is background? What is this blur and where is it originating from? So this background is actually a convolution of multiple different sources from the sample. They can be the out of focus signal, of course, which is a light collected from adjacent planes in wide-field microscopy. It can be autofluorescence of the sample, and it can also be detector noise. These are just a few examples. This results in an image with blur and a lack of contrast because our true signal is riding against all of this other background signal, which lowers our signal to noise. The goal of Thunder is to remove these out-of-focus sources and really give a higher contrast, better signal noise on the true signal. So on the right, we can see an example of what Thunder would be doing, where we remove that background, and now we have better signal and noise across our true structural details. So if we go back to that example of a tumor spheroid we saw a few slides ago, we see on the left we have our state-of-the-art fluorescence microscope wide field image. It's blurry, it has a lot of background, but we can see our structural details behind that. After Thunder, the background and blur is removed, we have better signal to noise, and we can see our structural details much more easily in the tumor spheroid. So how are we doing this? We're doing this with Thunder, which is our core technology, but then we actually have algorithms beneath that, one that is called instant computational clearing. And this is a proprietary technology from Leica, where we remove the out of focus light through our computational clearing method. So beyond it being the core technology of Thunder, it is meant to remove the wide field blur around a desired object size based on feature scale or feature size. It's meant to improve image contrast while preserving the spatial dimensions of the image and removing the inherent haze. It works on single images and does not require a volumetric acquisition. The resulting images are also available instantaneously, given the name instant computational clearing. So on the right, we see a raw image of an LG test slide. And on the bottom right, we see the ICC'd image. So we can see that the haze is removed and my structural detail is much more easily seen after ICC. So let's dig a little deeper into what instant computational clearing really is. So it's a feature scale based background removal method. It's independent of structural elements, discrete wavelets, and point spread function modeling. Instead, it's a mathematical model to distinguish signal, your true signal, from the background or out of focus light by implementing a cost function. So in the middle here, we have an example of what the equation may look like. This is a simplified version. We have a non-quadratic fit where you have your original image and the background, and then a regularization term for minimizing erosion of the signal. In our dialog box in the software, it's implemented with the lightning bolt, and you can set up parameters that we'll go through in the next slide. So to go a little deeper into that cost function, the minimization, and the non-quadratic. So Y is our input, which is our image, what we've collected in our wide field system. X is the background noise that's estimated to be removed, so that's going to be subtracted from the image. This gamma term is our regularization term, and it prevents overfitting and erosion of the data. Gamma itself is a regularization parameter that's proportional to the full width half max of the point spread function, but in our dialog box it's commonly referred to as a feature scale. This is a physical feature in your image that you would like to discriminate from out of focus and in focus, and it's what's used to decide what is out of focus in the image. Below that, we see strength. Strength is the amount of out of focus light that it tries to remove from the image. So you can set that between 0 and 100%. A little bit more about this dialog box. You can see that we can set up multiple channels, and we can actually go between channels and set different feature scales and different strengths. So you can really fine tune it to your sample. Going a little bit further, we'll give a few examples of how ICC is actually working. So this next slide is going to give us an example of how it removes the out of focus light. So again, we're going to be using that feature scale which is based on the structures that you've labeled with fluorescence. You set that here. The default value is proportional to the full width half max of the point spread function. So default will give you enhanced contrast just based on the point spread function. It's user settable. You can fine tune it per image, like I said before. But you can also save these settings and apply them across multiple images, so you're consistently removing out of focus light between images and samples. So if we start to set feature scales, we can explain how it's actually removing the out of focus light. So on the bottom left here, we see that we have these fine peaks across larger peaks and then an overriding large background. If I set my feature scale to be the width of this blue line, I'll remove the red line as that's determined as out of focus light. If I then set my feature scale to be the finest feature, the green line, the green peaks, it'll actually start to remove the blue line as well and give me a much flatter background across my true signal. If we take this and we do an example with an actual sample, say a whole mount retina, we can see here the raw image on the top left, our thundered image on the bottom left. We have a cross section and we have graphs showing what that cross section looks like. A little more about the sample. This is the red channel looking at retinal ganglia, and the red dots are the cells themselves. So if we were to want to quantify this, the raw has a very nonlinear background where it's going to be hard to quantify because it's not consistent. After thunder, it flattens and we still have our true peaks and our true signal, but it'll be much easier to quantify. So it instantly removes the non-homogeneous background. The post-thunder structures and relative intensities are preserved. And this new thunder data set can be quantified. So it can easily be segmented or have ratiometric analysis performed on it. So this is an example of what it does to the data. If we start looking at just larger images overall, we have an example here of chickling cochlea, where it's a tile scan taken with our 3D tissue imager. That's based on our upright. On the left, we have our Widefield. You can see all of the inherent haze in background. And on the right, you can see the image after thunder, where that haze is removed and we have better signal noise, higher contrast. Another example would be mouse bone. On the left is the raw. On the right is the ICC'd image. This is a maximum intensity projection, so you can see that we've taken a Z-stack, we've removed the haze, and then collapsed it to a single image to show you how the structure and the signal and noise has been improved. So to review computational clearing, it's a PSF independent process. It's complementary to your workflow and does have optional deconvolution, which will be discussed in the next portion. It's meant to be extremely fast, meaning we have that instant computational clearing. It's implemented on the GPU. You get your data instantly with acquisition. And here we just have a few more examples showing you how ICC improves image quality. On these adherent cells, the background is removed, the signal and noise is increased. On this mouse lung, this is actually a very hard image to collect because we used a 20x air through 1 millimeter of plastic, and you can see there's a lot of out of focus light, a lot of blur due to that. And after ICC, that is removed, and we have much better contrast and signal to noise across the features in the organoid.
As mentioned by James, the core technology of Thunder is instant computational clearing. ICC can be paired with adaptive deconvolution to not only improve the contrast of the image, but also the resolution. The deconvolution offered by Leica Microsystems is an accelerated adaptive Richardson-Lucy-like deconvolution that pairs with computational clearing. Its implementation is fully automated, and the deconvolution is adaptive to changing SNR's, signal-to-noise ratios, across the entire image. In fact, adaptive SNR coefficients are created and identified over the entire image. And this is shown here on the right-hand side, where regions of high SNR are identified as red, and regions of low SNR are identified as blue. And so the degree of deconvolution, as determined by the amount of regularization, is dependent on the local SNR in the image. The adaptive nature improves both the image contrast and the resolution. There are two methods for implementing this adaptive deconvolution, the first being small volume and the second being large volume. Both of these will be discussed a little later. The deconvolution is both regularized and constrained. Using a Bayesian statistics approach with a Gaussian noise process, the function to minimize for deconvolution is presented here, where H star O represents the convolution between the Gibson-Lonnie point spread function, PSF, with the object. The right-handmost term is a term that penalizes and constrains the deconvolution, and gamma is the regularization term that scales nonlinearly with SNR. On the right-hand side is the Thunder dialog box as presented in LASX. The top portion of the dialog box is similar to what has been shown before, and it's the parameters that govern computational clearing. You can see a variety of channels have been populated, and each of these channels can have a different feature scale value and strength value. And to remind you, feature scale is the size of the structures which your fluorophores are labeling, and strength is the degree of background that the user wants to remove. Below are the deconvolution settings. And as mentioned, deconvolution is implemented in an automated fashion. This means that the user does not need to select the number of iterations in order to converge the deconvolution. By using the auto feature, the algorithm automatically converges and identifies the ideal image that is the best deconvolved image. Another parameter is regularization, which, as mentioned, scales non-linearly with SNR. The default value for the regularization is 0.05. This is also user settable, and the user can adjust this value accordingly. I should point out that regularization is not a constant value. In fact, it ranges from 0 to 0.05. And what value is given in the equation depends on the local SNR. We also have an optimization feature, which is a list of Gaussian blurs with different kernel radii that is applied to the image prior to deconvolution. And this Gaussian blur functions as a way to frequency filter your dataset, where Gaussian blur acts as a low-pass frequency filter. As mentioned, adaptive SNR coefficients are created over the entire image. This results in the adaptive regularization to limit image noise and artifacts to preserve image detail. We're already familiar with the function to minimize for deconvolution. And as I mentioned, gamma is the regularization term that nonlinearly scales with signal to noise, or SNR. Here is the function that relates to SNR, where gamma max is the maximum predefined regularization. So based on the previous slide, that would be a value, or the default value of 0.05, and SNR max is the maximum predetermined SNR value. So putting this all together, if we consider this heat map, which describes varying levels of SNR throughout the image, where red represents high SNR, and blue represents regions of low SNR, adaptive SNR coefficients are created over the entire image. And the amount of regularization depends on the local SNR. In regions that have high SNR, the regularization value is low. In regions where the SNR is poor or low, the regularization value is high. As mentioned, there are two implementations of adaptive deconvolution. The first being small volume computational clearing, or what is known as SVCC. It combines ICC with adaptive deconvolution. And SVCC is ideally suited for thin samples that have low SNR, or signal-to-noise ratio. In SVCC, the adaptive deconvolution is performed prior to ICC, which means that the features of interest are enhanced prior to the removal of background. This is shown here on the right-hand side. We can see this raw image, and there's quite a bit of background in this image. And a lot of the features, while they can be identified, are in fact not very clear. But by applying the adaptive deconvolution, we improve the quality of the structures of interest, and also we remove the background. And so the result of SVCC is shown here. The other method for implementing adaptive deconvolution is large volume computational clearing, or what is known as LVCC. It also combines ICC, or computational clearing, with adaptive deconvolution. And LVCC is meant for samples that have high SNRs and who have background contributions that are common in thick samples. In LVCC, which is different from SVCC, ICC is performed prior to the adaptive deconvolution. This means the background is first removed, and then the remaining signal is treated through the adaptive deconvolution. This is shown here on the right-hand side. You notice that the raw image, the SNRs, are not that bad, and then we have quite a bit of background that overlays the image. By applying ICC prior to adaptive deconvolution, we remove this background and then treat the remaining features by the adaptive deconvolution. And the result here is shown below. Thunder can be performed on the fly, meaning during acquisition or post-acquisition. And it's important to mention that the raw data is always preserved. So you always receive a secondary data set, which is your Thunder data set. We also have a batch processing algorithm, which means that a series of images can be taken and processed post-acquisition using the batch processing algorithm, and such that the parameters for all of your images are the same in terms of the Thunder parameters. How does Thunder compare to other imaging modalities in respects to the size of the point spread functions? Here's a graph or a plot showing you the size of the PSF for Widefield and what the PSF would look like when we apply ICC. You'll notice that after ICC, the size of the PSF is indeed smaller. And this is a consequence of removing some of the background. But when we pair ICC with adaptive deconvolution, such as in the case of LVCC, we see a further reduction in the PSF. It's not quite as small as in other modalities, such as using confocal microscopy. And so if increased resolution is desired, Leica does offer confocal products, such as the Solaris platform, which can be paired with the deconvolution called Lightning, or you could use the STED 3X and the STED 3X with Lightning. Thunder is found on a variety of Thunder imagers or microscope platforms. Starting from the top left, we have the Thunder imager model Organism, which is ideally suited for imaging large biological systems such as zebrafish, C. elegans, Drosophila, or whole mouse. And the reason for this is there's a large working distance between the objective and the sample. We also have a family of upright microscopes like the Thunder Imager Tissue for slide work, And if you desire to take Z-stacks for some of your slides, we have the Thunder Tissue 3D, or it's the Thunder Imager 3D Tissue. We also have a family of inverted microscopes, the 3D cell culture, which can be enclosed with an incubation chamber to perform 3D live cell. And we also have the Thunder Imager 3D assay. So all of these systems are equipped with the Thunder technology. Throughout this presentation, we have shown you a few images of what Thunder can accomplish. And so we'll continue with that theme and show you a few more. Here's an image of a mouse lung organoid, imaged with a 20x air objective through 1mm plastic bottom dish, rendered in 3D. This is the image prior to Thunder, and this is the image after Thunder. Here is a mouse brain tissue stained with GFAP and 647. This is a 120 micron Z-stack. And this image is also rendered in 3D. This is the Thunder dataset. And that's the raw. Here's an image of a mouse lung stained for the AT1 lineage. This was a 70 micron thick Z-stack, also rendered in 3D. This is on the left-hand side, the raw dataset, and on the right-hand side is the Thunder dataset. And this was processed using large volume computational clearing, or LVCC. And lastly, here's an uncleared human brain organoid. It was a 2x1 tile scan. There was 4 fluorescent channels, and a total Z-stack thickness was 285 microns. And this image is a result after using LVCC.
So now we've discussed our Thunder technology. Let's see how it's actually applied to our instrumentation. We've given some examples, but we wanted to go through some specific hardware examples that you can use with the Thunder. So here we have our 3D cell culture. It's based on our inverted DMI8 platform. We can pair cameras, motorized stages, and other things with it to really give you a high-end microscope. So one of the biggest choices any researcher needs to make when doing microscopy is to choose the correct objectives. At Leica Microsystems, we have many different objectives to choose from. We have water immersion with a microdispenser for long-term imaging, motorized correction collars for doing multiphoton. We even have water or glycerol objectives to match refractive indices of your samples. We also have ones that go up to BAB and other types of clearing methods. We also have many with correction collars for allowing different thickness of sample holder to image through. So while we have all of these different options, it may be a little hard to find the one you need. So we've actually come out with what we call Objective Finder. And this allows you to search through our extensive catalog of objectives and really choose the objective that's correct for you and your application. So beyond objective selection, the other benefit you have from going with a compound microscope like the DMI8 is the ability to use real-time control of the system. So in many systems, when you have a USB controlled system, your illumination will turn on, it'll tell it to take the exposure, and then it will tell it to turn the illumination off. This time here is what's needed to communicate that back and forth in the system. When using a truly synced or synchronized system, like the DMI8 and our Synapse controller, when we tell the illumination to turn on and when to expose, everything is TTL triggered, so that our illumination is in line with our exposure and we are not overexposing our samples. So this is called Synapse. It allows for TTL triggering. We can do this with our stages, our LED light sources, and even third-party components so we can control things not sold by Leica as well. This allows for high-speed acquisition by synchronizing the camera with the illumination. This synchronization also allows for minimized light exposure, which minimizes or reduces photo damage and bleaching of the sample. Also on our DMI-8 is something that allows us to create and keep focus. We call this adaptive focus control. It is a way of locking the objective to the sample to minimize Z drift due to temperature fluctuations or mechanical drift. The way this works is that we have a far red LED light that comes in through the objective. So being far red, it does not expose your sample to unneeded light. It looks for the back reflection off of your sample. It works with air and multi-immersion objectives, so you are not restricted to air or oil. You can bridge all of those. It will lock your focus to the cell or your sample. And then as time progresses and you have fluctuations in the system, let's say your sample heats up a bit and it shifts the focus up higher. Without AFC, your focus would be now out of focus. But with AFC, the objective will follow that thermal fluctuation and keep the cell in your sample in focus. So an example of how this works over time, we have live cell imaging being shown here. So we're able to keep the cells in focus by having a rigid stand, but also that hardware called the AFC. We can also implement software autofocus as well. But what that really allows you to do is do long-term time lapsing. So you can see down here we have C. elegans embryos, and we're imaging those over the course of multiple hours. And we also have cell viability assays where we're imaging over multiple hours as well and tracking cell development and motion. So along with this high-speed imaging in this AFC, we also have stages that allow for multi-well plate imaging and give you high speed, more data points, and overall better statistics because now you have many more data points. So we can capture samples that go from either slides to well plates, so chamber slides, multi-well plates, things like that, through our custom carriers in our software. And this allows for automated imaging of those carriers where you're not setting up by hand a 96 well plate acquisition. With Thunder, we then have that available in real time through our computational clearing and adaptive deconvolution, where as we collect our data at high speed, we get our end result at high speed as well. So I've mentioned this custom carrier. This is all implemented through our software within LASX. It's called Navigator. This allows you to identify areas of interest within LASX. You can load and align and begin imaging with saved templates, which is being shown here on the right, where they're loading a template. And with that AFC and our other focus control methods, we can keep our sample in focus over these really large areas. So you can see he's implemented AFC here, and now as it goes along and images in each one of these chamber slides, the sample is in focus and he's collecting quality data. So with Navigator, you can get these very large mosaic images of tissue sections. You can also do multi-well experiments, multi-position experiments. So here is what we would do with a mouse slice or a slice of mouse brain, where we have our Thunder image on the left, our raw on the right. So this is a multi-tile acquisition where we've taken Thunder and then stitched it as a post-process. So to give you a few more examples of the kind of data you can get off of this system, we have here a transgenic prenatal 5-day mouse heart that's expressing GFP. And you can see that we can get a very large 3-dimensional image with our 10x objective using large volume computational clearing. And the full thickness of this sample is 740 microns. Another example using an organoid. This is actually an organoid cluster stained with DAPI for the nucleus, GFP for the plasma membrane. And you can see we can get the very large overall structure as well as all of the fine structure within that. And this was done with large volume computational clearing as well. Our next example is an adult mouse ovary that's been fixed and stained with anti-Minobox and then cleared with 3-disco. You can see that we have very clear image quality after deconvolution. This is a very thick sample taken at almost 600 microns with our 10x objective. The next example is a whole mouse embryo stained with beta-3 tubulin and cleared with BAB. And this is actually taken with a BAB multi-immersion refractive index matching objective, a 20x multi-immersion objective. So you can see we have a very thick section. It's 700 microns. It consists of 32 stitch tiles. And you can see before thunder, we have a lot of out of focus light. And then after thunder, our image quality is greatly improved with large volume computational clearing. The next example was mouse brain expressing AMPK in red and the microglia with IB1+ in green. It was taken with a 40x 1.3 NA objective, and both of these images are maximum projections taken from a 27 micron thick 3D volume. And you can see with the raw, we have a lot of out of focus light, as well as blur just from the inherent wide field imaging. After large volume computational clearing, we remove that out of focus light and enhance our image quality and resolution. This can also be applied to live cell imaging as well. So our next example is actually a 48-hour time lapse of live cells being imaged across multiple wells. This is an explant tissue of abdominal aorta. It's genetically expressing TD tomato, and when a transcription event occurs, it begins expressing EGFP. So in our magenta channel is our TD tomato, and you can see the cells that are in cyan, that is now our EGFP. And you can see over the course of many hours, going up to just under 48 hours, these cells can be tracked and distinguished between TD and EGFP.
As previously mentioned, thunder is included in our family of thunder imagers. Here's an image of the Thunder Imager 3D tissue. It's based on an upright platform. The 3D tissue imager is ideally suited for imaging your slide work. To see how thunder can benefit such samples, here are a few examples. Here is an image of a neuromuscular junction, immunostained for neurofilament and beta-bungarotoxin. The left-hand side of the image is the Widefield fluorescent image, and the image on the right-hand side is the image after thunder. This is a mouse lung stained for PHD, Psi3, and Alexa 633. The image was acquired using a 20x objective, and the final image was projected into extended depth of field, or EDOF. The left-hand side is the EDOF image, which is the wide field image, and the image on the right is the EDOF image after thunder. Here is an image of a Drosophila rendered in 3D within our LASX software. The stainings were 647 and 555, labeling a subset of motor neurons. Here is the image after thunder, and that's the image before thunder. The Thunder Imager model organism is a stereo microscope that is ideally suited for imaging whole model organisms, such as zebrafish, C. elegans, Drosophila, or whole mouse. To see how thunder can be utilized on such samples. Here again are a few examples. This is an image of a cortical spheroid, where red labels neurons and green labels astrocytes. The image was acquired using a 2x 0.15 NA objective set to a zoom of 3.4. The left-hand side is an EDOF image taken in wide-field fluorescence, and the image on the right-hand side is the EDOF image after applying thunder. This is an image of a C. elegans. The strain was MAH215 with a dual fluorescent reporter of M. cherry and GFP. The image on the left-hand side is the wide-field fluorescent image, and the image on the right-hand side is a result after thunder. Here we have an image of a sea anemone, stained in RFP and in GFP. Notice how you can clearly see GFP signal around the mouth and in bands in some of the tentacles. This is a result after thunder. Here is a mouse heart stained for Th positive neurons. It's rendered in 3D within our LASX software. The total Z-stack thickness was 400 microns. Here is the result before thunder, and that is the result after thunder. And lastly, this is an image of a mouse brain. The left-hand side is before thunder, and the right-hand side is after applying thunder. So hopefully with these images we have shown you, you now can appreciate the power of using thunder, the ability to see through the haze, to increase image contrast, and also image resolution.
With that, we'll finish on the last and newest member to the Thunder imaging family, the EM Cryoclem. So the EM Cryoclem is an upright-based microscope to allow you to image your samples at cryogenic temperatures and then do a coordinate transfer to your Cryo-EM or EM microscope to then do high-resolution imaging on those same areas. So the main benefit of the Cryo-EM is being able to determine sample quality and location before going to the Cryo-EM to do high-resolution imaging. So you can image across the grid and look at your sample quality, identify areas that are not good for imaging, like crystalline ice contamination, and then identify areas that are good and would be suitable for EM imaging. So you can see here we have intact meshes with our cells growing across them, good fluorescence signal. We could mark these and then take them to our Cryo-EM. So as mentioned, you can identify and mark potential targets. So here we have a fluorescence cell of interest. We could mark it as a coordinate. We could do this across the entire image or the entire grid. And then once we go to our scanning electron microscope or our Cryo-EM, we could do a coordinate transfer, find those exact same cells, and image them at high resolution, and even do tomography. Another large benefit is being able to maintain cryo conditions through all of this process. This maximizes your ability to successfully find samples of interest and get good experimental outcomes, while minimizing the potential of contamination during loading and transferring of your sample. This is done by keeping your sample at cryogenic temperatures. We do this with a cryogenic stage, an objective that works at cryo conditions, and then a shuttle that shuttle your sample at cryogenic temperatures between our system and your Cryo-EM. With this and with the fluorescence imaging, we've now added thunder to this as well. And on this next slide, you can see as we peel away, we have our raw versus our thundered image. So with the thunder, we get higher resolution, better image contrast, and overall a better, easier way of marking coordinates for transferring to the Cryo-EM. These are human cells stained with GFP and M-cherry, and we can target them now with coordinates that we could transfer to our Cryo-EM and then do high-resolution imaging across them. So that being said, we'd like to finish on two important parts of our Thunder imaging family. One being the publications that have come out so far for our instrumentation. Thunder was launched in 2019, and since then we've had over 60 publications using Thunder imaging. We've taken a couple selection from that. We have one based on cancer, looking at drug resistance in melanoma and Thunder imaging of cells. We have one in neurobiology, looking at membrane trafficking in brain slices using a Thunder imager. And then last, we have one in biotechnology, looking at conductive protein dye microfibers and using a Thunder imager to look at the fluorescence there. So we have this publication list available on our website, listed here on this link. If you'd like to find it by searching, you can search for Leica Microsystems Thunder's publication list. And the second piece we'd like to finish on is quantification. Always a major part in any fluorescence imaging is quantification. This is not an exhaustive look at quantification, but we'd like to give a few examples of how people can use our systems for quantification. This first one being counting. So this is a brain slice where we're looking at neurons in a mouse brain, and we want to count them. But we also want to look at the co-localization of our green and red channels. So this image on the bottom left is our brain slice, our thundered image. The image on the top right is of counting those neurons. So each one of these points is a neuron that has been counted. And then the image below is looking at the co-localization between the green-red channel, where we can now count when cells of two different colors are touching each other. Thunder is important for this because when we apply deconvolution, we're getting better image quality, but also better image resolution, which gives you better localization. You'll get better counting and localization with it. The second one we'd like to show is ratiometric analysis. This is of an Arabidopsis expressing GFP. As we apply a catalyst, peroxide, we can use our sensors to look at the response of the Arabidopsis. And in time, we can see how it responds to that injection of peroxide in the calcium expression channel. So this is a way of doing ratiometric analysis with the Thunder imagers as well. Again, not exhaustive, but it is a very important question that we'd like to answer and show. So with that, we'll finish with a review of the Thunder imager family. Again, we have three main instruments that it's based upon. Our model organism, which is on our stereoscope, our 3D tissue on our upright DM6, and our 3D cell culture on our DMI8. The main part, or the reason that we brought Thunder to the customer, is to allow for getting brilliant images fast. We also wanted to allow for effortless work with new models like organisms, tissue sections, and 3D cell cultures, and really allow people to take advantage of next generation disease models with not having a restriction to the hardware they have. So Thunder imagers are a new class of imaging system that rely on cameras for detection and high quality hardware, and Leica's computational clearing technology to remove out of focus light. And with all of that, we hope that you're able to see through the haze and see how Thunder can benefit you and your samples. To finish, I would like to give you an idea of who's been talking to you for the last hour or so. We have Vikram Kohli. He's the Midwest Advanced Workflow Specialist. And then you have myself, James Marr, the Mid-Atlantic Advanced Workflow Specialist. You can see that we have our emails listed. If you have any additional questions after this, feel free to reach out. We also have a remote application support line that you can contact, as well as a YouTube channel with tutorials on how to use our hardware and our software. And with that, we'll take any questions, and thank you so much for joining us for this webinar. Have a good day.
And thank you so much, Dr. Marr and Dr. Kohli, for that informative presentation. We will now start the live Q&A portion of the webinar. I would also like to welcome Levi Veltz, who will be joining us for the Q&A portion. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box, and we will directly go ahead and answer as many questions as we have time for. So let's get started. Levi, welcome. And it looks like we already have some great questions coming in. Our first question, you mentioned the deconvolution is regularized and constrained. What is the regularization used?
Hi. Yeah, thank you for these questions. We've got a few of them here in the chat, and I've tried to respond to some directly via the response, so hopefully you're getting those. Yeah, when we think about the regularization and constrainment of deconvolution and what does it mean, there are different methods that exist to regularize the deconvolution, to limit image noise, to limit artifact generation. Ultimately, the goal here is to preserve overall image details. So there are techniques like Tikhonov-Miller, total variation as a couple of examples that are used. But for Thunder, primarily we're using goods roughness as the regularization algorithm that's being used to help preserve these image details.
Thank you so much, Levi. And what does SVCC and LVCC vary in the order of deconvolution and ICC?
Yeah, so if we remember back, SVCC is our small volume computational clearing, LVCC is large volume computational clearing, and ICC is interactive computational clearing. And each one of these can be done independently, but and it depends on a lot of different factors. Sample thickness is a primary factor. The signal to noise ratio is a primary factor. The amount of background presence. So if you're measuring a thick sample with a lot of background, deconvolution on its own can fail to produce a restored image. So what we would use would be the large volume. And so large volume is going to do computational clearing first and then apply deconvolution. But if you have a sample that maybe there's poor signal to noise or it's very thin, you would want to do deconvolution first in the workflow and then do computational clearing. And that's where you'd use small volume computational clearing. So there's a lot of different factors that go into which one that you're choosing: thickness, signal to noise ratio, background. Most of the samples we're looking at, and we've seen people use, the large volume computational clearing is the default mode that they'll go to. But if you're looking at something that's very thin or has a very weak signal to noise, you can switch modes, and all of the modes are, you can try them both. Because it's done computationally, you can try one, you can try the other and see which is giving you the result you're looking for. And then there are other optimization parameters you can use in the software to really customize the experiment to fit your the analysis to fit your experiment.
Thank you so much, Levi. And our next question from an audience member: can Thunder be applied to BF images?
So yeah, BF. So what we're talking about is bright field. And in short, the answer is no. So Thunder is really applied to the fluorescent channels. So within our software system, LASX, for people using Leica microscopes, if there is a multi-channel acquisition setup with a bright field channel, the bright field channel is skipped as part of the algorithm. So we're only applying we're only applying thunder imaging computational clearing to the fluorescence channels.
Thank you so much. And again, I want to thank our audience for these great questions. We have time for one more question. Is the data quantifiable?
So James mentioned this on the last couple of slides, and quantifiability again is a variable in the community that we work in. So we can think about, is it quantifiable from a segmentation analysis perspective? Is it quantifiable from a fluorescence intensity measurement, ratiometric analysis, all of these things. But post-Thunder, the data sets can be quantified for segmentation and cell identification and cell counting, fluorescence intensity, and we can preserve all of these relative fluorescence intensity. So yes, it is quantifiable, and one of the key aspects we have with our Thunder Imager platform is you always have the raw data to look at and compare to. So when you're measuring peak heights or what's changed, we don't overwrite the raw data. We don't delete the raw data. The raw data is always there, and then the computational clearing and the thundering of your samples are all done, as a separate module to work with. So you can always compare and contrast. But yeah, those are all good. There's still some other questions in the chat. We'll take these questions down and we'll email you directly the answers, because I know we're running towards the end of time here. But these are all great questions.
And thank you so much, Levi, for your participation with the Q&A. And I also want to thank Dr. Marr and Dr. Kohli for their time and for their important research. And I remind audience that questions we do not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the registration. I'd also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. You can view the webinar on demand, and LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, take care, everyone. Bye-bye.